Bismillahirrahmanirrahim. Assalamu alaikum dear students. Welcome to the new lecture. Today our topic is another second step of the aerobic respiration, that is Krebs cycle. The Krebs cycle is also known as TCA cycle, which stands for tricarboxylic acid cycle, or CAC cycle, which stands for citric acid cycle. Now coming into definition, so we can define Krebs cycle as that the cyclic process in which the high energy acetyl-CoA molecules are completely oxidized into carbon dioxide and water is called Krebs cycle. The cyclic process in which the high energy acetyl-CoA 2 carbon molecules are completely oxidized into carbon dioxide and water is called Krebs cycle. Now discovery. Krebs cycle was first discovered by a British biochemist, Sir Hans Krebs, in 1937 and therefore named after his name as Krebs cycle, for which he was awarded with Nobel Prize in 1953. Right? Location. Where does Krebs cycle occur? So Krebs cycle, occur in mitochondria, in mitochondrial matrix, where all the necessary enzymes are present. Now coming into the mechanism or process of Krebs cycle, so we will explain the process or mechanism of Krebs cycle or TCA cycle or CAC cycle with the help of this diagram, right? Now it's a closer view of this diagram or process, that is mechanism or process of Krebs cycle. We will start from linked reaction. Again, a revision for you. If you remember from the last lecture that as you know that pyruvate molecules, 2 pyruvate molecules are produced at the end of glycolysis, and you know that pyruvate is a 3 carbon molecule. So during the linked reaction, you know that the pyruvate does not enter the Krebs cycle directly, and during the linked reaction, this pyruvate, which is a 3 carbon molecule, is oxidatively decarboxylated. Oxidatively means that the pyruvate is oxidized, that is hydrogens are removed from this pyruvate molecule. These hydrogens are taken up by the oxidized NAD, which get reduced into NADH2, while decarboxylation means that carbon dioxide is removed from the pyruvate molecule. So with the removal of or with the decarboxylation, the pyruvate is now changed into a 2 carbon molecule, which is called acetyl group. The acetyl group attaches with coenzyme A. The coenzyme A attaches with this acetyl group, making a 2 carbon molecule known as acetyl-CoA. This acetyl-CoA then now enters the Krebs cycle. And this process is called linked reaction because it links glycolysis to the Krebs cycle, right? So this is a brief revision of linked reaction. Now this is a Krebs cycle. If you look into this diagram, the Krebs cycle completes in 8 steps. Step 1, 2, 3, 4, 5, 6, 7 and 8. We will start right from the step number 1 with a closer view. So now in the first step of the Krebs cycle, the acetyl-CoA, a 2 carbon molecule produced in the linked reaction, combines with the pre-existing oxaloacetic acid or oxaloacetate. The acetyl-CoA produced in the linked reaction combines with the pre-existing oxaloacetate or oxaloacetic acid, a 4 carbon molecule, in the presence of water molecule, forming a 6 carbon molecule known as citric acid. The acetyl-CoA 2 carbon molecule produced in the linked reaction combines with a pre-existing auxiliary 4-carbon molecule in the presence of water molecule, forming a 6-carbon molecule known as citric acid. Acetyl A, CoA, it is a 2-carbon molecule, while oxaloacetic acid or oxaloacetate is a 4-carbon molecule. So they combine. So these two combine in the presence of water molecule, forming a 6-carbon molecule. 1, 2, 3, 4, 5, 6. This is known as citric acid. While the coenzyme A, if you look into the diagram, is now becomes free and is ready to react with another acetyl group, right? So this is the first step in Krebs cycle, and this first step takes place in the presence of an enzyme, citrate synthase, while the linked reaction takes place in the presence of an enzyme, pyruvate dehydrogenase. Pyruvate dehydrogenase, right? Now in the second step, in the step 2, this citric acid is changed into another 6-carbon molecule known as isocitrate in the presence of an enzyme known as aconitase, right? So citrate or citric acid is changed into isocitrate. Before moving into the next step, now why Krebs cycle is also known as CAC cycle, citric acid cycle? So the reason is that, as you know now, that citric acid formed in the first step, citric acid is an early intermediate of this cycle. The citric acid is an early or the first intermediate of this cycle, so that's why the Krebs cycle is also known as citric acid cycle, right? Now in the third step, the isocitrate, this 6-carbon molecule, is oxidatively decarboxylated, just like linked reaction. Isocitrate is oxidatively decarboxylated, which means isocitrate is oxidized, that is, hydrogens are removed from isocitrate, which are taken up by the oxidized NAD, and this NAD is reduced into NADH2, right? While decarboxylated means carbon dioxide is removed. So when carbon dioxide is removed, so this isocitrate, a 6-carbon molecule, now changes into a 5-carbon molecule known as alpha-ketoglutarate. Alpha-ketoglutarate. So in the third step, isocitrate is oxidatively decarboxylated in the presence of an enzyme, isocitrate dehydrogenase. Oxidatively means isocitrate is oxidized, hydrogens are removed. These hydrogens are taken up by NAD, which get reduced into NADH2, while decarboxylation means carbon dioxide is removed. So the isocitrate, a 6-carbon molecule, is now changed or converted into a 5-carbon molecule known as alpha-ketoglutarate. So isocitrate is oxidized to alpha-ketoglutarate. 1 carbon of isocitrate is oxidized to carbon dioxide, and hydrogen is removed, which is picked up by NAD, reducing into hydrogens are removed, which are taken up by the NAD, reducing into NADH2, right? Now coming into the fourth step, now this alpha-ketoglutarate, which is a 5-carbon molecule, is again, now this alpha-ketoglutarate in the fourth step is oxidatively decarboxylated, which means that oxidatively means that ketoglutarate is oxidized, that is, hydrogens are removed, the hydrogens are taken up by the oxidized NAD, which get reduced into NADH2, while decarboxylation means another carbon dioxide is removed. So with the removal of this second carbon dioxide from alpha-ketoglutarate 5-carbon molecule, it is changed into a 4-carbon molecule known as succinyl, right? This reaction takes place in the presence of enzyme alpha-ketoglutarate dehydrogenase. So as you are looking, this is the second carbon dioxide molecule produced in the Krebs cycle. 1 carbon dioxide molecule in this third step, and now this second carbon dioxide molecule in the fourth step, right? So the alpha ketoglutarate is now changed or converted into succinyl group. The coenzyme A, if you look into this diagram, the coenzyme A reacts with succinyl group. This coenzyme A reacts with succinyl group, forming succinyl-CoA, right? Now coming into the fifth step. In the fifth step, the succinyl-CoA is converted into succinate. The succinyl-CoA is converted into succinate, a 4-carbon molecule, and coenzyme A is released. The coenzyme A is now released. And some of the energy produced in the oxidation, is used in the synthesis of ATP. Some of the energy produced here in this oxidation is used in the synthesis of ADP, from ADP and inorganic phosphate. So once again, in the fifth step, the succinyl-CoA is changed or converted into another 4-carbon molecule known as succinate. The coenzyme A is released, and some of the energy produced in this oxidation is used in the synthesis of ATP from ADP and inorganic phosphate. As the energy of the substrate is used in the generation or synthesis of ATP. As here the energy of substrate, this energy comes from the substrate, from the oxidation of substrate. So the energy of substrate is used in the synthesis or generation of ATP. So this synthesis of ATP is called substrate level phosphorylation. This phosphorylation is known as substrate level phosphorylation. Why it is called substrate level phosphorylation? Because the energy of the substrate is used in the synthesis or generation of ATP. So that's why it is called substrate level phosphorylation. If you recall from the light reaction, there we studied photophosphorylation, the synthesis of ATP from ADP and inorganic phosphate using energy of sunlight. Using energy of sunlight, that is called photophosphorylation. But when using the energy of substrate, when this energy comes from the substrate molecule for the generation or synthesis of ATP, then it is called substrate level phosphorylation. SLP stands for substrate level phosphorylation, right? And remember, this fifth step takes place in the presence of an enzyme known as succinyl-CoA synthetase. Succinyl-CoA synthetase. Now you must know that in plants, the succinyl-CoA, this enzyme succinyl-CoA synthetase, in plants, this succinyl-CoA synthetase enzyme produces ATP. It produces ATP molecule. While in case of animals, the GTP molecule is produced. As you know that ATP stands for adenosine triphosphate and GTP stands for guanosine triphosphate. Guanosine triphosphate. ADP, adenosine diphosphate, and ATP, adenosine triphosphate. GDP, guanosine diphosphate, and GTP, guanosine triphosphate. So remember that this enzyme succinyl-CoA synthetase in plants produces ATP, while in animals it produces GTP, guanosine triphosphate, GTP, right? However, in animal mitochondria, ATP may be formed. Later on, ATP is formed in a second reaction that involves the transfer of phosphate from GTP to ADP. However, in animals, in a second step, in a second reaction, ATP is formed. How ATP is formed? By transferring the phosphate from GTP to ADP, right? Now coming into the sixth step. In the sixth step, the succinate is oxidized into another 4-carbon molecule known as fumarate. Oxidized means hydrogens are removed from succinate, which are taken up by oxidized FAD, which get reduced into FADH2, and this oxidation of succinate occurs in the presence of an enzyme, succinate dehydrogenase. Now seventh step. In the seventh step, the fumarate is changed or converted into another 4-carbon molecule, malate. In this step, the fumarate is changed or converted into malate. How? The fumarate is hydrated, that is, water molecule is added, so it is converted into malate in the presence of an enzyme, fumarase. In the eighth and last step, the malate is now oxidized into oxalocitrate, that is, hydrogens are removed from malate, which are taken up by oxidized NAD, which get reduced into NADH2, and the malate is oxidized or converted into oxaloacetate, and a molecule of NADH2 is produced during this step. This oxaloacetate or oxaloacetic acid is now able to react with another acetyl-CoA molecule and continue the cycle. So these are the 8 steps of the Krebs cycle or citric acid cycle. Why it is called citric acid cycle? Because citric acid is produced in the first step, right, which is a 6-carbon molecule, citric acid. And why the Krebs cycle is called TCA cycle, tricarboxylic acid cycle? So the reason is that the Krebs cycle is also called TCA cycle. TCA stands for tricarboxylic acid cycle. So it is also called TCA cycle because each molecule in the Krebs cycle contains 3 carboxyl group. 3 carboxyl group are carbonyl carbon. Now this is the overall equation of TCA cycle or Krebs cycle. As you know that this is pyruvate molecule and there are 2 pyruvate molecules produced at the end of glycolysis. So overall 4 NAD molecules. These pyruvate molecules are oxidized into 3 molecules of carbon dioxide. 1 carbon dioxide is produced in the link reaction, while 2 carbon dioxide molecules are produced in the Krebs cycle, right? 4 NAD molecules are reduced into 4 NADH2. 1 NAD is reduced in the linked reaction, this one, and the 2 and the 3 NAD are reduced in the Krebs cycle. That is in the step number 3, 4 and 8. 1 FAD molecule is reduced into FADH2 in the step number 6 of the Krebs cycle, and 1 ATP molecule is produced in the Krebs cycle in the step number 5 through substrate level phosphorylation, right? And 2 water molecules are also produced. So this is the overall reaction of the TCA cycle. As you know that there are 2 pyruvate molecules, so thus in all, if you multiply it by 2, so thus in all 6 CO2 8 NADH2, 2 FADH2 and 2 ATP molecules are produced in the linked reaction and Krebs cycle. And for one glucose molecule, the Krebs cycle occurs twice because there are 2 pyruvate molecules produced at the end of glycolysis. So this is the TCA cycle or CAC cycle or Krebs cycle. So with this we complete the TCA cycle or CAC cycle or Krebs cycle. Thank you.